discussion of fluorescence. And then we talked about DNA structure. Oh, okay. Guys, I'll be on YouTube. You could be too if you come up to the stage for some demos. So. I brought my Feynman lectures and other textbooks to demonstrate different sizes for different bases. A and G, they're bigger than in guanine, and they are called what? Purines, shorter, right? And then C and T are smaller, just like my Shirovich quantum physics textbook, and they are called pyrimidines. And then when you put two of them, pairing like this, that's base pairing. But real interaction that stabilizes DNA, double helical structure, is actually the stacking interaction between the adjacent bases. And this is really a hydrophobic effect. You don't want to have this hydrophobic, aromatic rings next to water, so you want to shield them as much as possible. And then stacking one against another really helps you achieve that, right? Yes?
When you say aromatic rings, I've heard that description, aromatic, a number of times regarding chemicals. What does that actually refer to?
You got me there. Okay, let me guess. Okay, so whenever you have a ring, and then you have seen structures like this, right? So you have double bonds, but actually double bond does not stay in one place; they move around, right? It's resonance. So you have more delocalized electrons instead of staying in one place. Okay, and so that's what I think that aromatic means. But actually, anyone actually has a better idea? It must be called aromatic because many of the chemicals smell, right? I don't know, maybe caffeine has the structure too. So I'm sorry, I'm not a chemist.
It's okay.
It's okay.
Thanks.
Yeah. But that's what I mean. I don't know whether that actually is true. What I mean by aromatic rings is these kind of ring structures. This hexagon, a pentagon.
And a floating bond.
Yeah.
Okay, thank you.
And the fluorescent dye molecules have multiple rings like this. And so electrons can be delocalized once you excite it, right? Then, shuttle, like this. Just like electrons oscillating in a TV antenna.
That would make for a stronger dipole response.
Yes, I think so. So today I want to continue the discussion of DNA structure, and then I want to talk about two-state dynamics, just using a DNA hairpin structure as a specific example, but more in general terms. And then I want to talk about how mechanical force can change the dynamics and give you information about some of the hidden structural features. Okay, and then finally, I want to talk about DNA mechanics. Just to let you know, so I'll give another lecture on Thursday. Next week, I will be out of town. My postdoc, Jingyi Fei, will give, actually, so what she'll do is on Tuesday next week, she'll give 1 hour, which is the regular lecture on FRET and super resolution imaging. And then on Thursday, you will come to my lab. There will be 2 students doing hands-on demos with you guys, actually analyzing some data too. And so it'll be divided between FRET and super resolution cellular imaging. And 5 will start with FRET, and the other 5 will start with imaging, and then you will actually switch in between. Okay, so Jingyi will tell you where to meet and so on Thursday. But it'll be actually interesting to see how these kind of measurements are being done. Questions? But that was a really good question. Aromatic. All right, DNA structure. So that is a ribose ring. So that is actually part of the DNA structure and essentially a sugar ring. And in the chemical structure, my understanding is that whenever you don't see any, it's kind of, bodies here, that means you have a carbon atom there, unless it's marked. So I put an oxygen atom here, so the oxygen, but here carbon here, and then DNA. So for this ribose group, there's a numbering for the carbon. This is carbon 1. This is carbon 2, 3, 4, and then there's another carbon here, and then it connects to PO3, phosphate, negatively charged, and then it connects to third carbon of the next unit. And then this guy connects to PO3, phosphate, and then it connects to another carbon, and then and so on. So that's still 1, 2, 3, 4. And this carbon is called number 5. Okay. And where do you attach the base? You attach your base here to first carbon position. And this base can be A, G, with the purines, and T or C. All right, let me tell you one thing. So this is called a 2' position. And there, basically what you have a carbon, and then there is a hydrogen there. Okay, that's what it is. This is for DNA, but for RNA, instead of having a hydrogen, you have a hydroxyl group. OH. So for RNA, the difference is that you have OH. Then you make an RNA. Okay. So this is RNA, and then for DNA, okay. So when you make a molecule like this, it's called RNA, which is ribo nucleic acid. What is DNA? DNA is deoxy ribo nucleic acid, right? So name really comes from the fact that, compared to RNA, DNA has one less oxygen. So if you start with the RNA, sugar ring, and then remove oxygen, deoxy, then you get the DNA sugar ring. Okay. So in DNA and RNA, actually, now the research is so specialized. If you go to a meeting specializing in nucleic acid in general, then DNA people and RNA people form different camps. Okay, so when DNA people have a session, then RNA people go out, skip the session and go out and have lobsters, and things like that. So DNA people, RNA people tell DNA people, oh, we are studying the real thing. You guys are studying modified RNA. Okay, in a sense, it's true, right? Maybe especially if the life form started from the RNA. Right? So in fact, there is a fake Genesis written by somebody. I don't know who wrote it, but in the beginning there was light, whatever, in the Bible. But in the beginning there was RNA, and then God thought it was too lonely, and then removed the ribose, from where, and then you made DNA. You made oxygen from RNA, and then you made DNA, and it's like that. Anyway. So another thing that I want to mention is that this actually is a very stable structure. What makes RNA unstable is actually just this one, 2' hydroxyl group, because this is highly reactive chemically, and it can be actually cleaved by itself in high pH and so on. And this is a reason why some people never believed that RNA could have been the beginning of life, because it's not stable enough. But this is a reason also why RNA can actually function as an enzyme. So actually, you cannot make an enzyme made of DNA, but you can make an enzyme out of RNA, really because of this. Okay, it's highly reactive. That means you can also use it to do some catalysis. Questions? Have you heard of 3' or 5' DNA? Do you know where it comes from? So these are called 1', 2', 3', 4', 5'. Okay. So from the viewpoint of this ribose ring, this is a 3' direction. That is 5' direction. Okay. On this strand of DNA, that's 5', 3'. So there's a polarity. It's not symmetric along this direction. There's a polarity. That's what they call 5' and 3' direction. I have a friend in Paris, and he published a paper in Physical Review Letters, and he wrote in the figure caption that he used 3' DNA strand, whatever, right? But the Physical Review Letters, they have editing staff who actually they spell out acronyms. So, for example, if you write AFM, then it can mean one of the two things, antiferromagnetism or atomic force microscope, and they just expand without really looking at the context. So you can get misleading information. And then, do you know what HF is?
Fluorescence.
Huh? There's a chemical, hydrofluoric acid. Right? Did you watch Breaking Bad? That's where the guys use hydrofluoric acid to, dispose human remains, right? But anyway, so there was one paper which was originally written saying that we etched the sample using HF, right? But then the editing staff expanded this without asking, so it became they etched the material using Hartree-Fock. Hartree-Fock is actually an approximation used in physics, right? So anyway, so they expanded this without asking, so it became 3 foot DNA, and actually makes sense because I told you that if you measure the entire length of the 23 chromosomes you get from your mother, it's about a meter, about 3 feet. So anyway, so that's just a side story. So when you have a double-stranded DNA structure, you can actually write one strand as an arrow, and this is. Sorry, I switched my convention here, direction here, but the pointed end is a 3' direction, and this other one is a 5' direction. And when you form a double helix using two DNA strands, they don't run parallel. They don't run in the same direction. They actually run in opposite direction. So DNA has this anti-parallel structure. So what it means is that once you form a double chain DNA, it's symmetric. There's no defined direction. Question?
So what defines the direction between the 3' and the 5'? Is it that the proteins that like polymerase tend to go one direction?
Yes, exactly. Yeah. So motor proteins moving on DNA, many of them are moving on single-stranded DNA, and they move only in one direction, either 3 to 5' or 5 to 3'. Just like kinesin, dynein can move one direction, right? Depending on which protein you have. So interesting question. For some proteins, let's say this is like 5' direct. This direction is defined direction, and then you have proteins that move in this direction, right? And then you have proteins moving opposite direction. Then how do they do this? Do they bind in the same way, but simply take back steps like this, or do they bind in the opposite direction and then move forward? Okay? And so far, it looks like they actually bind in the same way, but for the molecule moving in the other direction, they take back steps instead. Do you know how to build a DNA molecule? How to build a DNA molecule?
So we already have a DNA molecule to start with?
Yeah, then in that case, yes.
Okay. Then all you really need to do is unzip it gradually with one set of protein machinery, and then another set of protein machinery creates an environment where single base pairs can come in and join, and then you ratchet to the next base pair location, and the next one happens in and joins, and then you ratchet to the next location, right?
Yes, exactly. That's how it works. So let's imagine that you have a DNA structure like this. So you have a single-strand DNA region, and then enzyme called a DNA polymerase can come and bind here, and then add one at a time, one unit of DNA at a time. It's called actually a nucleotide. So that's one unit of DNA. And then you add one at a time. It can be A, C, G, T. And then once you add this one, then it becomes one nucleotide longer. And then enzyme moves forward by one unit and then continues to add. Okay. And this is a very accurate process, including all the error corrections. Maybe you make one mistake every billion reactions. But the DNA polymerase has to start with what's called a primer. So this is a template strand. And then you need to have a primer for a DNA polymerase to function. If you give just one strand of DNA, then a DNA polymerase cannot start. Okay, so this raises an interesting point. If you have a really, really long DNA, okay, and then as it is, there's an arrow. So which direction is this? 3'. And 5'. So if you have a primer somewhere here existing, then the copying can occur in which direction? So DNA, the polymerase always copies in the 3' to 5', 5' to 3' direction. So every known DNA polymerase copies in this direction. So then this guy will, copy DNA in this direction.
Wait, I thought you said it was 5 times 3 prime.
The only made strain is 5 to 3'.
Oh, all right. Okay.
Okay. And then you may also be able to do this starting from here, right? And then, but because it cannot make DNA in this direction, right? So there's a problem of actually how to copy the end of the DNA. Okay. So there's a fundamental limitation in molecular biology because every DNA polymerase can copy in this direction. Everyone requires a primer. Then there's an issue is how to copy this one. Okay. So actually you can't. You cannot copy that, right? So if you copy the DNA and then you lose a bit here, and then you divide the cell and you do another replication for the daughter cell, and you lose a little bit more here. So then as you go down in division cycle, as you make more and more cells, genomic DNA becomes shorter and shorter.
Will be back on itself. 3' to the 5'.
Okay, the nature has solved all these problems, just like 2 ways you suggested. First, in bacteria, DNA is not linear. DNA is circular. Okay, so you solve the problem by making a circular genome. So E. coli has a circular genome. But human DNA is not circular, it's linear. In human DNA, we have some special structures at the end. Okay, and that's called telomeres, yes. And topic of another Nobel Prize, like 3 years ago. And 2 of them who got the prize were professors when they made the discovery, but another one was a grad student of one of the professors. And I said what they discovered was not the telomere, but they discovered an enzyme called telomerase. And I don't like the name because it doesn't conform to my expectation that it has to be named after whatever is destroying. But in this case, it actually is extending a telomere. Okay, what is a telomerase? Telomerase is actually upregulated in cancer. Meaning that in cancer cells, you have a lot of telomerases, meaning that telomerase protein expression is upregulated. Okay? Why do you need to do that? Because cancer cells divide quickly, right? And if the cancer cells keep losing the end of the DNA each cycle, then they will no longer be cancerous, right? They can divide a little bit, and then because of defects caused by the loss of DNA in the end, then basically cancer cells will just stop dividing, and then it's not tumorous anymore, right? So cancer cells have to upregulate the telomerase to actually extend, so to this end of the DNA. Okay. So that is a telomerase. So naturally, many companies have been studied to find drugs that can inhibit telomerase. Then these cancer cells will not divide anymore, and then maybe you can cure your cancer. So far, no one has been successful, but many companies have been studied, and their stock price went up and down, depending on big paper coming out in some journals and so on. All right, so that's one way of making DNA, right? If you have a template DNA, and then you can use a DNA polymerase and a primer to actually copy the DNA. Another way of making DNA is to buy it. Okay? Actually, you can make DNA chemically. Okay? That the way it's done. So there's a very there's a big company in Iowa called IDT DNA. So actually, I think a majority of people doing research buy their DNA from this company, and you can actually open a web browser, and then you can type your sequence. I want to have a DNA that has into their web browser, and then you enter your credit card number, and then they will make it for you and then send it to you. Okay, and for simple ones, it can be just $10. Shipping is more expensive actually. We have to be careful because it's fully automated. So as soon as you press the button, then the machine will start making it. You cannot actually cancel that, right? So some students cry in my office after making that mistake. No, just kidding. So the way this kind of approach is made actually is done actually now in the opposite direction. Okay? So what they do is that they have large surface, like bead surface, and they start with 3' nucleotide, let's say G. That's how you start with. And then, but the end of the G is actually there's a protection group there. And then you deprotect using some chemicals. And then you add the next one. But this also comes with a protection group so that it doesn't actually add more than one. And then you wash everything away. You deprotect, and then you add the next one, and so on. So that's how you grow the DNA from one end, going up one at a time. The chance of making a mistake or getting termination is about 1% on average over many sequences. So typically, if you make 100 nucleotides long DNA chemically, half of them will be too short to be useful, and so on. But that's basically how you build DNA chemically, and I'm sure you know forensic activities where you can study DNA, right? But without actually a chemical way of synthesizing DNA, you would not be able to do any of those measurements. So this is really enabling technology. Okay, questions? All right, so next I want to talk about DNA hairpins. So let's imagine you have a DNA sequence that starts with ATGG and then TTTTTT, many T's. That's the convention. You write down the sequence from 5' end to 3' end. Okay. Then what can happen is that these guys can form a hairpin you have ATGG pairing with, this is 5' end, 3' end, TACC, and then you have, so that's the DNA hairpin. So if you want, it kind of looks like that. This part is called the stem of the hairpin, and this part is called the loop. When you have a stem as short as 4 base pairs, this is not that stable, so it will most likely, melt within less than a millisecond. But then, it has nowhere else to go, so it will actually rezip. Okay, so this can actually go back and forth between the loop state. Between the two states, reversibly. So this is called the closed state. This is called the open state. And okay, if you increase the temperature, which do you think will be preferred? Open structure. Why? Entropy. That's a great point. So, nature wants to minimize free energy G, right? Nature wants to minimize free energy, and that is there's enthalpic energy minus, what is it? Ts, entropy S times T, right? So if there's no enthalpy, then nature wants to maximize entropy here, right? So of the two states, which do you think has a high entropy? Open, because it's more disordered, right? High entropy state is basically college students' room, right? And this is the room of a graduate student. Right?
Maybe a graduate student without children.
Okay. Anyway, so when you increase the temperature, contribution of distance becomes more important. So then whatever has a higher entropy will win, right? So that's why this is preferred at high temperature. If you increase the salt, like magnesium and sodium in solution, then this state becomes favored, because here negative charges that you have negative charges everywhere, every base here, every nucleotide, and the ions in solution functions to screen these interactions. So if you have higher ion concentrations, then the closed state is actually more favored, and so on. Okay. If you have more GC base pairs, which would be favored? Closed, because of because we have more hydrogen bonds? No, because second interactions stronger if you have more GC base pairs. Okay, why do you care about DNA hairpin? Actually, my answer is that no, actually you don't need to care about DNA hairpin, because DNA really doesn't exist in this form in the natural state. Okay, only in very exceptional case you'll see a hairpin like this, because DNA is always made, starting as a template and then, one at a time. right? So it's always more or less existing in double-strand DNA. You don't see that. You need to care a lot about RNA hairpin because RNA is actually single-stranded when it's made. It doesn't form these extensive double-stranded structures. So, one reason why we may want to study this is because DNA is a good mimic of RNA, and it's easier to handle and so on. So if you have a piece of RNA, then it will also form this type of what's called the secondary structures. So this is the primary. So if you want, this is a primary structure. What I mean by primary structure is simply, the sequence of the DNA. Okay, and something like this is called the secondary structure. So it utilizes the internal interactions to form higher level structures. The RNA also can form secondary structures. So if you go to Chicago airport, I do that all the time. And then that's terminal, here is your terminal G and whatever. Terminal So in concourses, you have all these interesting gauge and structures. So RNA secondary structure looks just like this. You have this type of hairpin and loop, and another hairpin and loop. Okay. So RNA can fold into actually fairly well-defined secondary structures based on the sequence. Okay. So when you have closed and open such a two-state system, you can write down the coupled differential equations to describe the kinetics. So you define P open as the probability that you find the molecule in the open state. Okay? And then you can go back and forth between the closed and open state with certain rates. So I use K to denote a rate. Unit is second, inverse second. And I'm going to call this K opening, the rate of opening the hairpin. I'm going to call this K closing. And probability that you are in this open state is P open. This is P closed. So you can write down a simple equation here. Change in the open state probability is given by how much you had there to begin with and how quickly it decays to go to the other state, and how much you had in the other state and how quickly it comes to the open state. Likewise, what am I doing?
Yeah, so you want to write P closed, I'm pretty sure. Or,
Okay. Okay, so it's about the simplest possible coupled differential equation you can write down. And in equilibrium, that means population is already has reached equilibrium. It doesn't change as a function of time. This means that these values are zeros. So this is 0. That is also 0. So then what you get is the ratio between the closed and open state is simply given by the no. So you can think of some type of reaction coordinate. And let's say that the distance between the two ends of the DNA is x. Then this guy can you can have two minima, one for the closed state, and this is for the open state. For the open state, that end-to-end distance will be longer, right? Larger. So that will be consistent with this idea. So this will correspond to closed hairpin. This will be open hairpin. So if you come here and then measure G of closed, and then measure G of open, so free energies of the closed and open states, then the ratio between the two will be given by from Boltzmann distribution. And the energy difference between the two will be the delta G. So relative populations between the closed and open states is entirely determined by the free energy difference between the two states. And if there is no free energy difference, then this will be 1. They have, equal population. So, but it is actually more interesting to think about how quickly they move, not just the ratio between the two, but to think about the rate of transitions. Because, in biology and life, changes are very important, and some how quickly they can go back and forth is not determined by the free energy difference between the two states. It's determined by the barrier height. Okay? So if the barrier is higher, then thermal energy is not enough to allow the molecule to climb up the barrier and then cross over to the next one, and so on. So depending on how high the barrier height is, the reaction can be much faster or much slower. So that barrier height is typically written as delta G dagger. So rate of opening, how quickly you can go from here to there, can be generally written in this form. Prefactor, and it has a unit of dimension of second power, and then so how quickly a reaction can occur is determined by the barrier height. Okay. So one way of thinking about this is the following. A can be thought of as attempt frequency. So if you have a ball sitting here in this potential well, and the thermal energy will allow it to move around. Okay. And then at this frequency, and I just measure, okay, how much instantaneous energy do I have? If the energy is actually less than this barrier height, then I don't make a transition. But if that instantaneous energy turns out to be higher than this, and then I make the transition. Okay. So basically, you check that energy with this frequency, and then your probability distribution of that energy follows this, and if you have enough energy, you move over. So that is how you actually can actually think about the rate of transitions in this formulation. Now, for very simple chemical reaction, very, very simple chemical reactions, you can say A is equal to the thermal energy over the Planck constant. And if you do the calculation, this is about 10 to the 13. So that will be about once every 100 femtosecond or 0.1 picosecond. Basically, this applies if you have a very simple atom. So a molecule, let's say you have a diatomic molecule in vacuum, right? In that case, you can use this expression, and that's called, this becomes Eyring's equation. So what it means is that you look at this diatomic molecule, and then every 10 femtosecond you examine how much energy it has obtained from the thermal fluctuations. If the energy is now larger than the barrier, then you move over. If not, you stay. Okay. So that is the formula that you can use, and it works well for very simple molecules. But it doesn't work for biological molecules really, because the molecules are much more complex, okay? And they're in water and so on. So what it means is that even if you can measure this, you cannot determine what the free energy barrier is, because really, in most cases, you don't know what prefactor A is. Okay? So people studying protein folding, they make a living by arguing about, well, I'm saying too much, but about how to estimate the Freenet barrier, because you cannot measure it directly based on the rate alone. One more thing that I want to mention is that this state right at the barrier is called the transition state. And it is by definition a transient state. Like, who has seen the wind? Neither.
Seen the what?
Seen the wind? Right, okay. It's a poem, okay. But it's, no one has seen the transition state. That's what I want to say, okay? Except in computers, in the computer simulations. And one way of defining that transition state is that once the molecule goes to that state, then you have 50-50 chance of coming back to this state or moving over to the other state. So in computer simulations, people actually use that criterion to actually discover a transition state. Doing lots of simulations, and which of the reproducible state that you see in computer simulations actually have a 50-50 chance of going back to the original state versus to the other state. And so delta x here is called the distance to the transition state. And one more thing, this x is called the reaction coordinate. And so in this case, the end-to-end distance of the hairpin is named the transition state. And this kind of makes sense intuitively. But what it really means is that the fact that you can describe the system in this one-dimensional coordinate assumes that aside from this lengths, all other degrees of freedom are quickly averaged out compared to the relevant time scale via experimental analysis. So that is usually the assumption that this is a key reaction coordinate, and every other degree of freedom is averaged out rapidly. Okay? And in this case, is very likely. Once you hold this DNA at a certain distance, then it'll undergo fluctuations, but quickly averaged out. So you only need to look at one coordinate. And this type of analysis is also called transition state theory. And Zubair, who is in Caltech, he won the Nobel Prize technically for having detected transition state using femtosecond lasers. Although everyone knows that the prize was given to honor the development of ultra-fast lasers, but they had to give it to one person, and they had to give a reason why that person, and then they picked a paper where they actually showed evidence of a transition state using fast laser-based spectroscopy. Okay, questions? Yes?
In general, can we know or at least guess at the structure of A, if not the actual numbers associated with it? Because if you could, then you could vary T, for instance, and gain a knowledge of delta G, just by knowing how A and E to the whatever vary with T.
Yeah, so obviously if you assume this, the A depends on T, right? But if you look at this, the rate depends much more strongly on T through this term, right? So this term become pretty negligible in the big scheme of things. And there are other formulas. Instead of identity, there's a theory called Kramer theory. And in that case, you have a more complex term in front. People argue about what those mean and what the values are for the different elements in that formalism. But in the Kramer theory, you assume that this actually it diffuses along this reaction coordinate. Okay, and but really, it's not. I don't think it's really. There's a really accepted way of actually calculating the prefactor or structure. There are competing theories, and Kramer theory is the most commonly used one. It also has a temperature there, but again, not as strongly dependent on temperature, as in the exponential here. Any other question?
So for when can we assume A equals to ABC by A? So you have said very, very simple reactions. So that would mean only like single step reactions, like first order reactions? Or
Okay, for example, can we use this term for DNA hairpin? No. Okay. And essentially, any kind of mechanism like proteins and DNA, I don't think you can use it. And some people use it, but that's not. I don't think that's correct. Actually, we tried to use it, and then we got scolded by a reviewer like 10 years ago. So, then you're not supposed to do that. Okay. And yeah. But a lot of physical chemists, they use it for fairly complex reactions too, right? So I don't think that's correct. Okay, let's take a break. Oh yeah.
Is the prefactor expected to be different for the different rates?
For different processes?
Well, for K close to equilibrium, would you expect it to be very different? So it seems like if you could measure both of those and the relative ratios, that'd give you delta G and then two equations.
The relative ratios will give you delta G.
The delta G, free energy. And then if you can measure both K's, then you don't know, then you could figure out what A and delta G plus plus are, because you've got two equations and two unknowns.
No, you cannot.
Do you expect them to be different, or?
No, even if they are the same, even if you know that they are the same, that's not going to give you the barrier height. Yeah, if you come up with a way to do this, I think you'll become famous. Yeah. You may actually win the big prize. Let me know. Maybe you can also include me in your. Did you know this story about a professor who comes to the lab, in the afternoon once a day, and then ask his student, okay, what have I discovered today? All right, any other question? Okay, let's take a break, and then we'll come back for another 15 minutes. So I told you that a lot of people care about the nature of the transition state, and it's a human nature that you like to get or know what's not possible to get or difficult to get. So people want to know the nature of the transition state. You want to know the structure. You want to know the structure of the molecule, transition state, molecule in the state, and so on. And it's hard because it's by definition very short-lived. So one hint one can imagine is that if you can somehow find out what the distance to a transition state delta x is along that reaction coordinate, then you may be able to, restrict the range of models that are possible for that structure of the transition state. So information about that distance can be useful. Okay. For example, you may have a transition state that looks like this, or that looks like that. Okay. So for something like this, this is the hairpin that you have, right? And then here is that there's a small separation to the transition state. In the transition state, you may have only, so instead of having 4 base pairs, you may have only 3 base pairs, like this. And then here you have a fully melted one. Okay? So if you were to actually determine that delta x is very small, you may be able to conclude that this is the transition state structure. Okay? Whereas if you find that delta x is very large, as shown here, then you may conclude that that is the transition state. Okay? So it's actually useful to determine this distance to the transition state. Now, how do you do that? We can actually use the mechanical force to achieve this. Imagine that, in some magical manner, you can grab 2 ends of DNA hairpin and then apply force, magnitude of F. Okay? Then, which of the 2 states will be preferred?
The open.
Open structure. Why?
Because, well, this mysterious force is granting you an extra benefit to your energy the farther apart the 2 ends are.
Exactly, right? So like if you have a gravitational force pointing downward, then the ball wants to be down there, not up here, right? Because potential energy is actually minimized by going down there, right? So what you can think of this is, so when you apply force, then what you're doing is that you're actually applying additional potential energy to the system, so that when x becomes large, then you have actually a lower energy. Okay, nature wants to keep things in the lower energy, so basically force will prefer, in a conformation state with larger distance. Okay, and that slope will be in terms of free energy, extra free energy that you get is actually F times X. Okay, so what you're doing is that you are adding this this much energy, linear energy term, to this, right? So what happens is that after you have done that, then you'll be actually moving things like this. Okay, and here same. You are adding extra potential like this, so you are moving this. So something like this. And notice that because of small separation here, the reduction in the barrier height, because coming from this extra potential, is not very much here, right? What it means is that when you apply force, the force-dependent change in the forward reaction is actually not very big. Whereas force-dependent change in the reverse rate will be much larger. Does that make sense? So because of small separation here, force has a much larger effect on the backward reaction, closing reaction, than in the forward reaction. Likewise here, the force will have a much larger effect in the forward reaction rate than the backward reaction rate. Okay? So so if you were to measure the rate of opening, okay, and then plot in the log scale, and as a function of force, opening will actually increase, and with force, and the log scale is a line. That's because, let's say initially I had a barrier height of delta G dagger, but then new barrier height after I apply a force will be zero force barrier height minus F times delta X. So I'm reducing that barrier height by this much. Then, because the opening rate here is as a function of force is and I can rewrite this as K opening zero force opening rate times okay. That means I can write log of K opening as okay. This is a reason why delta X. This is, sorry, I forgot delta X. This is a reason why when you plot opening rate as a function of force, it actually is a line, okay? And then from the slope, you can determine delta X. Okay? If the slope is large, then delta X is large and vice versa. Okay? So the idea here is that by performing like the measurements as a function of force and how the rate of reaction changes as a function of force, you can actually plot this in the log scale and then get the delta X value. So that is the idea. Okay. And people have done this to death now, by now. I mean, it all began about 10 years ago from a group in Berkeley. But then now, people have studied a zoo of DNA hairpin structures, now RNA hairpin structures, using this approach. Now, how do you actually achieve this? You can actually use a method called optical trap or optical tweezers. And this is a method invented by Art Ashkin in 1986. And so you can use focused laser beams to trap small dielectric particles about a micron in size. And if you attach one of those particles here and another particle here, then you can, using 2 laser traps to hold them and apply force and measure the distance between them precisely. So you can actually measure how the distance between the two is changing, closing and opening as a function of time and also as a function of force, and you can do all these measurements. So I think sometime in November, there's one additional guest lecture by Jan Schemner. So he's going to introduce you the technique of optical trap and its applications in biophysical problems. Questions? All right, so now I want to talk about DNA mechanics. Yeah, I don't have too much time, so we'll just start. Okay, so how many base pairs of DNA does your cell have? Single cell of your 3 gigabase pairs. That's from your mother, and then also you get 3 gigabase from your father. Then you have 6 gigabase pairs. Okay, so that's about 2 meters total. And how about in E. coli? E. coli bacterium living in the gut. Guess. More or less? Less. Okay. How about rice genome?
I think E. coli would have less than rice.
Compared to human?
Rice would be less.
I don't know, it might be comparable.
I think it's more. The genome is longer in rice. So E. coli has about 5 million base pairs. Okay, how long is that?
Well, if we have 6 billion and it has 5 million, it's like a third of a foot.
Third of a foot?
2 millimeters. 2 millimeters?
That sounds about right. 1.5 millimeters, yeah.
Oh, whoops.
So let's say 1.5 millimeters for E. coli's genome. And how long is E. coli? So E. coli kind of looks like this, right? Fluffy Cheetos. And the length is about 3 microns. This is about 1 micron. So DNA is actually much, much longer than this. So for the DNA to go into the E. coli cell, then it'll have to be compacted by some means. Okay. I will not do the calculation, but well, actually, let's not do this now. But There's another genomic DNA called lambda DNA. Lambda DNA actually is taken from a phage called lambda phage. Phage is a virus, and phage is a virus that infects bacterium. Okay, and in the case of lambda phage, its genomic DNA is about 50 kilobase pairs, 50,000 bases, so which is like even shorter than the E. coli genome, and this is about 16 micrometers, fully stretched. Okay. Now, DNA has a property in such that, if you have 16 micrometer long DNA, establishing DNA, you let it, be in solution. It does not actually stay in this stretched conformation. It actually collapses. It becomes like spaghetti ball. The average size becomes about 1.5 micrometers. Okay. And that we can calculate next time. In contrast, if you have a microtubule, we discussed a microtubule is a track on which kinesin and dynein molecules move as motor proteins. And if you have a microtubule of this length, then actually it has that length. It doesn't actually coil up to become flexible. It has enough rigidity to maintain its direction for a much longer distance even then. And actin filament is somewhere in between. Okay? So, and there is a number called persistence length to parameterize these properties of so-called biopolymers, and we'll discuss that next time. Thank you.